thanks to Kai for willingness to give a talk today, and I thought I'd quickly introduce him. So Kai is originally from Singapore, but he did his undergraduate degree at the University of Sydney, including an honors project that involved a lot of museum specimens, and this project was on the taxonomic revision of Stripes using both morphological and molecular approaches. Kai is very active on social media, so you may have crossed paths before. His work gets a lot of media coverage through outlets like the New York Times, Forbes, ABC News. In the years that he's been working on fish taxonomy, he's developed a lot of partnerships in the aquarium industry. He has a highly regarded track record for his career stage, so he's got over 20 published articles to date, a number of new species descriptions. Some of the species may figure prominently today in his talk. And he was also awarded the Australian Museum Research Institute Postgraduate Fellowship last year. And so part of the talk today is going to be about some of the work that he's done on that. And he's based at UCEs, co-supervised by professors Simon Ho, Nate Lowe, and Tony Gill at the Macleay Museum, as well as myself. So thanks very much, Kai, and I pass it to you, and I'm going to mute my microphone now.
Thank you very much, Joey, and the Australian Museum staff for having me today. Like Joey said, today I'll be talking to you about a project that was partially funded by the AMRI Postgraduate Award that involves the phylogenomics and evolutionary history of the fairy wrasses from the genus Cirrhilabrus. So before I get into that, I just want to give a quick run through on the background of the family. So fairy wrasses come from the family Labridae, commonly known as wrasses. They are among the most charismatic groups of fishes on the coral reef. They are the 5th largest fish family overall, and the 2nd largest in the context of the marine system. They're a cosmopolitan family. They're found all over the world in all sorts of habitats, but their diversity reaches the maximum within coral reefs. And they're not only known for their wide variety of coloration and form, but also in their functional specialization and the ability to exploit novel trophic goods. Basically, if there's anything that can be eaten on the reef, there is a wrasse that specializes in eating that. And they have a huge and very rich taxonomic history dating back to the 1800s. And over the years and decades, it's been changing quite a bit. But till today, the relationships of the wrasses are still very unsatisfactory. And this ranges from a contested monophyly of the family because of the nesting of parrotfishes to intrafamilial contention, such as some tribes being paraphyletic and even some genera being both para and polyphyletic. Now, I'm particularly interested in this group of wrasses informally known as the pseudocalines. They are represented by 6 different genera. We have Cirrhilabrus, also known as the fairy wrasses. We have Paracalinus, the flasher wrasses, Corneella with only 1 species, known as the mutant wrasse. We have Pseudocalinus, the mystery wrasses, and then we have 2 lesser known genera, Pseudocalinops and Pterygogus. These are all fairly small diminutive wrasses that are found exclusively on coral reefs, and they are either plantivorous or they feed on benthic invertebrates. Unusually among wrasses and all fishes, they have a really characteristic eye morphology in that the scleral cornea is bifurcated and bisected into 2. This is a little side project that I'm working on at the moment and trying to figure out why this eye morphology is such. And previously, the pseudocalines were thought to be the sister group of the calines, but more recent molecular studies have shown that it actually occupies a separate lineage in the wrasse family, distant to most other species. And it's a sister lineage to an assemblage of other labrids. And within the pseudocalines, the fairy wrasses are of the most systematic interest. There are 61 valid species, making this one of the most speciose genera, and they account for nearly 10% of all extant wrasses. They are very colorful, live on coral reefs at depths between 10 to 250 meters. And although all wrasses are sequential hermaphrodites, it is within the fairy wrasses that this is the most conspicuously displayed. The males are the ones that are more colorful. So all the photos today that you'll be seeing are of the males. The females they're still colorful, but considerably much duller. And they engage in very beautiful and elaborate courtship displays, flashing iridescent colors. Here's an example of one of the species. This is Cirrhilabrus taminkii. And on the left, you can see in its resting colors, it's very pretty. But when it's in nuptial display, it's a totally different fish. It's brightly colored and iridescent. And I just want to show a really quick video of them doing this. So that's a male courting a female. And so you can see the females are a bit duller, and the males are the ones that are doing this really cute little dance where they flick their tails to show the brilliant metallic sheen. And they live in big groups where the males are greatly outnumbered by females. This is also one of the more taxonomically advanced groups, for so to speak, where there are new species literally being described every year. In 2016 alone, there are 4 species. This year, there has been 1, with many more to come. So a genus that is continuously and still currently expanding. But despite this big systematic interest in the fairy wrasses, their evolutionary relationships have been very difficult to resolve, leaving a lot of open questions concerning their diversification and their evolutionary success. Previous studies have relied solely on mitochondrial DNA, but this has led to limited success in resolving the relationships of the fairy wrasses. This in particular stems from this phenomenon called haplotype sharing, where species that are morphologically or biogeographically separated share very conserved mitochondrial DNA. To make matters worse, they're not very morphologically informative as well. A lot of the characters, although very character-rich, are either apomorphic, homoplastic, or prone to intraspecific variation. So they're not phylogenetically informative. These factors combined have made the fairy wrasses one of the more phylogenetically recalcitrant group of wrasses. So here's an example showing what we call haplotype sharing. You can see here on both panels a bunch of fishes that look very, very different from each other, but we see basically no horizontal branching at all. So they all have identical mitochondrial CO1 sequences in this case. And this can happen based on 2 processes, what we call incomplete lineage sorting, where a particular gene has retained ancestral polymorphism and over time has not been able to sort into their distinct species, or as a result of mitochondrial introgression, which is caused by hybridization. And so these 2 processes are very hard to tease apart because they exhibit very similar phenomena, but the underlying processes are inherently very different. On top of that, we also have a bunch of wrasses, species that are what we call inserte status. And that just means of enigmatic phylogenetic placement. We don't really know where they belong. We don't know what they're related to. They're just kind of oddballs floating around in the tree. So we have 3 of the fairy wrasses here, Catoi, Labridae, and Claire. And then we also have Corneella here, which is a really unusual fish. It lacks pelvic fins and all associated internal pelvic elements. And I'll talk more about this guy in a little bit. But yeah, so all these together have made the fairy wrasses one of the more intractable groups to resolve. They also have really interesting biogeography. So the genus attains a widespread distribution across the Indo-Pacific. And I've just color-coded the different regions here to reflect biogeographical provinces. But although the genus is very widespread, they have an asymmetrical distribution where almost 60% of the species are disproportionately centered around the coral triangle. And as we move away from this central region, species diversity drops precipitously, with only 2 species in the Red Sea, 6 in the Western Indian Ocean, 1 in Hawaii, and 3 in the Southwest Pacific. So yeah, really large range, but really proportionately distributed. So for this study, we know that mitochondrial DNA is not very useful and morphology is also not very useful. We wanted to do a kind of whole genome approach in terms of tackling this problem. And so for this, we use something called ultraconserved elements, which are really short, highly repeated regions of the genome that are characterized by having a central core that is very hyperconserved. But as you move away from this core region, you get increasing sequence variability. So it's a combination of this core sequence and the variable sequences at the flanks that provide informative phylogenetic signal. And it's been shown that UCE data in the past with other vertebrate groups, including fishes, are very good in resolving evolutionary relationships across both deep and shallow timescales. The one caveat, though, is that because the core regions are so conserved, UCEs individually may not have enough signal to resolve relationships. So bearing all of this in mind, we then sought to, number 1, resolve the evolutionary relationships of the fairy wrasses using a combination of UCE loci and mitochondrial CO1. We wanted to then estimate the divergent times and reconstruct the historical biogeography of the fairy Wrasses, excuse me. And then also we want to compare different methods of phylogenomic inferences based on two methods, a summary coalescence method and a concatenation method. With very big data sets like this, especially with molecular sequence data, we wanted to make sure that the method we chose reflects the best biological sense, so to speak. And so I'm going to skim through the methods really quickly, but basically what we did is we sampled 39 of the 61 species for target capture of UCEs. We then assembled two supermatrices, one with a 75% occupancy totaling to 330,000 nucleotides, and another 95% occupancy matrix totaling about 15,000 nucleotides. We then fortified this UCE dataset with mitochondrial CO1, bringing the total taxon coverage to 80%. So for the mitochondrial DNA, we added species that were already found sampled for UCEs, but we also wanted to include stuff that UCE didn't sample for, but that represented key species complex. Basically, we wanted to have coverage of at least every major Cirrhilabrus species group, just for adequate representation of the genus. We did 3 different types of phylogenomic analyses. The first was a concatenation analysis using both maximum likelihood and Bayesian inference. Basically, what this does is you stick every single UCE end to end to form a mega alignment, and then you analyze it as a single data set. We also did a multi-species coalescence, which operates on a slightly different principle. So here we assume that each one of the UCE loci has its own genealogy, and then we infer an overarching species tree based on a coalescent approach. And we also did ancestral range reconstruction and molecular dating using secondary fossil calibrations from previous Labridae studies, and inferred biogeography using biogeobears. This is a very complicated slide, but basically the gist of it is that the summary coalescent trees failed to resolve the relationships of the fairy Wrasses. Even when we removed gene trees with poorly supported branches, even when we added CO1, and even when we only removed the most informative UCE loci. 4 of the species complexes were consistently recovered as para or polyphyletic. But in contrast, concatenation of the UCEs yielded a much better result, with all trees being monophyletic for the entire genus and also for all the different species complexes. We found that inclusion of the CO1 alignment to the UCE alignment did not affect overall topology. Instead, it allowed for better representation of all the unrepresented taxa. So this is the best resolved tree that we have at the moment of the fairy Wrasses, and this is a combination of the 75 UCE occupancy matrix and the CO1 alignment. And basically, this circular tree at the top here is an old tree that we did using mitochondrial DNA. And you can see here, the problem with the haplotype sharing is that all of these species have very little horizontal branching. But with our current UCE dataset, we have managed to resolve all the tips for all these different complexes. And so now they're all, there's a lot of species resolution now in our tree. Yep, so the concatenated UCE for CO1 matrix resolved relationships for the fairy Wrasses. We also resolved the positions of several of these taxa that were inserted status, such as Cirrhilabrus padui, Labridae, and Claire. We also found a really surprising result in that Corneella, the mutant Wrasse, is deeply nested within Cirrhilabrus. And this is reflected in every single one of our trees, coalescent or concatenation. So I don't want to touch too much on these plots, but basically what we have here is a RF matrix just to show congruence in tree topology. Basically, it says that every single one of our concatenated trees have very similar topologies, whereas the trees that were inferred using the coalescent approach were wildly different between all of them. And we think that this is due to the low information content of each individual UCE alignment. And so we did a average bootstrapping approach for every single one of our UCEs, and we found that on average they do indeed have a much lower bootstrap value. So it's likely that the summary coalescence was ineffective due to the low information content of each individual UCE locus. And here's a summary of our main results. So Cirrhilabrus is a sister group to the fairy Wrasses, Paracalinus, and the successive sister genera are Pseudocalinus, Pseudocalinops, and Pterodobus. Corneella is nested deeply inside Cirrhilabrus, and there are a total of 14 species complex from 7 major lineages. So here again, we have resolved the placements for some of these tricky taxa, and those that had intractable mitochondrial haplotypes have also been resolved. So I want to touch really quickly on Corneilla, the mutant wrasse. This was erected on the sole basis of lacking pelvic fins and all associated osteology. And on the top here, you can see a photo of Corneilla, the external morphology, no fins. Here's a cleared and stained Corneilla that I've inverted the colors just to show the lack of pelvic fins. And this lack of fins is quite rare in perfoid fishes, and within the wrasses, only one other species from one genus has this. But we show that in all our analyses, Corneilla is deeply nested within the fairy wrasse, indicating that this loss of fin is a derived trait. So it's essentially just a really strange and unusually abnormal fairy wrasse. And we think that this is unlikely to have been adaptive because Corneilla lives in an area where several other fairy wrasses also live, and they all share the same habitat type, so there is no real need for this loss of fin. But we speculate, or we hypothesize rather, that this is a result of population bottlenecking. This is a phenomenon where you have a species living in a really, really narrow range, and in this case, Corneilla is only endemic to a small cluster of reefs off the western coast of Western Australia. And if you have a really small population, your effective population size goes down, and if you have any deleterious mutations accumulate in the population, it could be fixed really easily, leading to permanent fixation. And this is quite parsimonious because we have found normal fin-bearing species of fairy wrasses to lack pelvic fins. So we know that this is a naturally occurring congenital defect. And you can imagine for a fish like Corneilla with a really small population size, this kind of mutation can be easily fixed and made to be permanent. But this is all just kind of, we just hypothesize this process, and we still need to do more investigation to kind of tease apart this mechanism. You also notice that it has a really uncanny resemblance to this one other species, Cirrhilabrus earli, in external morphology looking almost identical, except that it doesn't have any pelvic fins. However, the relationships of both species are not well resolved in our tree, and they have really unusual purple staining bones and scales. So if you put any of these species in alcohol, where you would have coloration normally stripped off, some of these scales and even the craniofacial bones develop a really intense purple staining, and we still don't know why this is so, but we think that this is a potentially plesiomorphic character because it's found in only one complex of fairy wrasses, but at the exclusion of two species. Anyway, so with our UCE tree, we decided to do molecular dating using fossil calibrations. And we found that the crown age of the pseudocalines emerged around 44 million years ago. The split between the fairy and the flasher wrasses were estimated at around 12 million years ago, with the crown group of Cirrhilabrus emerging shortly after at 9 million years ago. And the diversification of the fairy wrasses can be broadly characterized by two major events. The first, characterized by deeper radiations during the Miocene between 5 to 7 million years ago, where you have the emergence of all the major species complexes, followed by a much shallower radiation during the Pliocene-Pleistocene boundary, where all the species complexes started emerging somewhere around 4.5 to 2 million years ago. We also did a biogeographical ancestral range reconstruction. So we estimated the ancestral range for all the fairy wrasses as Melanesia and the central Indo-Pacific. So if you look at the map above, that would be the purple and kind of brownish orange region. So the ancestral range overlaps quite nicely with the area of the highest diversity for fairy wrasses. And we found 5 independent colonization events into the Eastern Indian Ocean. So the Eastern Indian Ocean is marked by this kind of moderately middle blue color. And anywhere on the tree where you see this at the nodes indicate an incursion into the Indian Ocean. 3 of these lineages further dispersed into the Western Indian Ocean, and of these, 2 of them moved into the Red Sea. So the movement of fishes from the Pacific to the Indian Ocean is a very well studied phenomena. It's been demonstrated very well for several other different taxa. And we know that the Indo-Australian archipelago is a very feature for this incursion from one ocean basin to the other. It's because unlike some of the other ocean basins like the Atlantic and the Eastern Pacific, this region is not characterized by any hard barriers. What this means is that there is no impenetrable barrier. Rather, the barriers are ephemeral and episodic due to all the little islands, especially during the Pliocene or the Pleistocene, when sea levels were rising and falling. These created transient barriers. So if you look at the map again, here is a map of the Indian and Pacific Ocean with the red triangle highlighting the coral triangle. Most of the fairy wrasses are found here. This yellow dotted line represents the boundaries between the Eastern Indian Ocean and the Western Pacific, or what we know as the Indo-Australian archipelago. And during the Pleistocene, all these little islands would have been submerged when sea levels were rising, allowing for movement of fishes from one ocean basin to the other. And in several of these fairy wrasses, they continued the dispersal into the Western Indian Ocean and then into the Red Sea with only 2 representatives. So this general pattern is what we believe to be responsible for the movement of fishes from one ocean to the other. So this leads us to the vicariance in species pump hypothesis, where the emergence of fairy wrasses within the coral triangle during the Pliocene and early Pleistocene overlapped with episodic lowering and rising of the sea levels during interglacial periods. And so these barriers, although very numerous and frequent, were often not very long lasting, and so occurred only over short spatial and temporal scales. So this, as you can imagine, will promote rapid diversification, but only on a very localized scale. And because they're ephemeral, they present very few opportunities for widespread, sustained dispersal, which is why we think the fairy wrasses have a really disproportionate geographic distribution across the Indo-Pacific. And this is characterized by rapid diversification on a very small scale with very few opportunities for widespread movement outside of the central area. But, you know, what about vicariance? It obviously, or not obviously, but we think that it has to occur as well, because if you look at these 3 species, they are very closely related, they look very closely related, and they have geographical distributions that are kind of like a jigsaw puzzle. So, you know, they conform to well-known biogeographical regions. So we believe a combination of vicariance and dispersal has shaped the movement and evolutionary success of the fairy wrasses, although teasing apart the nuanced mechanism is not very easy at this moment. Here is another really interesting group of fairy wrasses with a very interesting biogeographical distribution. So this is Cirrhilabrus exquisitus, and it's a single species, but it's characterized by half a dozen or more polymorphic forms that correspond very, very nicely to well-known biogeographical regions. And because it spans the entire range of the genus from the eastern coast of Africa all the way to the French Polynesian islands, we don't really know if this was a single widespread species that then kind of got broken up by vicariance, or if there was a dispersal event that moved them from one region to the other. We didn't really have very good text on sampling for this, so we couldn't really answer that question, but it's something to think about and something to investigate further. And another thing that we kind of touched on briefly, but was not really part of the main study, was the possibility of sexual selection influencing phenotype and genotype discordance. We believe that the strong sexual selective pressure exerted by female mate choice has led to the rapid evolution of morphological diversity and reproductive isolation of the fairy wrasses. And this is very well known for other groups of fishes, such as hamlets, and sexual dimorphism is a very important factor in courtship and nuptial display. And these sex-specific color patterns are sometimes, especially in the fairy wrasses, the only way of differentiating congeneric species. So if you look at Caribbean hamlets, for example, you have a species complex where they all look super different, and they all have very conserved mitochondrial DNA, just like the fairy wrasses. But on the genomic basis, it's quite a homogeneous landscape with very isolated peaks corresponding to visual equity, color interpretation, and color production. So we think that this sexual selection driving speciation also occurs in the fairy wrasses, but this is very difficult to test, and it's not well executed in the context of marine systems. But in our fairy wrasses, for example, we have one group that is that demonstrates this pretty nicely, and it's the Cyanopura complex. And this is represented by 8 or 9 different taxonomic entities that are clustered around, again, the coral triangle. But a lot of them have like completely overlapping distribution. They're entirely sympatric within their range. And as you can see, this is also reflected in the mitochondrial DNA, where there is basically no resolution at all. The whole thing is just a big mess of identical sequences, but yet they all maintain very, very distinct coloration pattern. And the lack of intermediate phenotype suggests that there is some sort of selection that's driving these evolutionary processes and preventing species erosion. And true enough, our UCE phylogeny have been able to resolve the relationships of all these species, suggesting that there is something else that's driving the selection of these fishes in a landscape where biogeographical and mitochondrial homogeneity exists. And in this particular group, they are very well known for their strong biofluorescence. And we think that a combination of environmental light properties, body coloration, and spectral sensitivities is what's driving, what do you call it? Genetic isolation, in that they only breed amongst themselves. Yep, and again, just to reiterate the point, the lack of intermediate phenotype suggests that there is a lack of species erosion and that the evolutionary trajectories of these fishes are being driven and maintained by other selective forces that we don't really know yet. Okay, so I'm just going to end the talk here with a bunch of conclusions. So we demonstrate the capacity of genome-wide UCEs in their ability to resolve phylogenetic relationships, especially in a recently diverse group like the fairy wrasses, characterized by very few morphological characters. Our results indicate that a summary coalescent approach is unable to resolve the relationships because of fully resolved individual gene trees that show very low information content that confirms previous assertions that the concatenation approach is preferred over the summary coalescent when estimation is the dominant cause of gene tree incongruence and not incomplete lineage sorting. And although the fairy wrasses emerged within the Miocene, their evolutionary success was largely driven by rapid cladogenesis during the Pliocene and Pleistocene due to the formation of all those barriers and them being centralized within the Indo-Australian archipelago. Yeah. So, okay, I'm just going to end this here. I'd like to thank again the Australian Museum for partially funding this with the Postgrad Award, and also all the partner institutions for lending tissue samples and letting me examine material. I'd like to thank Amanda Hay, Sally Reeder, Karen Parkinson, Joey, and all the other co-authors involved in the study as well. All right, thank you.